Welcome to today's webinar focused on innovative approaches in precision medicine oncology. I'm Eric Gifford from Collaborative Drug Discovery, and I'll be your host for this session. I'm delighted to be joined by three distinguished experts in the field. Today, we'll explore IDEAYA Biosciences' groundbreaking work in synthetic lethality and targeted therapeutics discovery. Before we begin our discussion, I'd like to quickly go over a few housekeeping items. For any product-specific questions, please use the Q&A tab in the toolbar. I'd like to invite our panelists to introduce themselves and their organizations. Dr. Tamsin Mansley, would you please start?
Sure, thank you, Eric. Yes, my name's Tamsin Mansley. I'm president of Optibrium Inc. and also head of Optibrium's global application science team. At Optibrium, we develop elegant software solutions for small molecule design, optimization, and data analysis. We're a science-driven company, and our research and development is into the models and algorithms that go into the software, helping to guide scientists' decisions in drug discovery. We have 4 platforms forming an integrated ecosystem. Today, we're going to focus on Stardrop, which is in use here at IDEAYA, but we also offer 3 other platforms. Sorella surfaces transformative insights revealed from your data and using AI-guided discovery. Our Biopharmics platform is for fast, robust 3D molecular modeling from small molecules right through to large microcycles. And Sameta provides accurate metabolism insights really tailored to the needs of drug discovery DMPK scientists. I'm particularly excited to discuss IDEAYA's innovative oncology research, Mick, and contribute to today's conversation. So why don't I hand it over to Mick?
Hello, everyone. I'm Michael Kapler. As Tamsin mentioned, people call me Mick. We're a precision medicine oncology company focused on synthetic lethality, an approach to target cancers with specific genetic mutations. Our platform combines computational methods with experimental validation to identify novel therapeutic opportunities in cancer treatment. And I look forward to sharing our perspective on how this approach is transforming oncology drug discovery.
All right. Hi, everyone. I'm Janice Darlington. I'm a customer engagement scientist at Collaborative Drug Discovery. So prior to joining CDD, I worked as a research scientist specializing in biochemistry and molecular biology. Been in the industry for 27 plus years. At CDD, I leveraged the scientific background to help research teams implement data solutions that enhance their discovery workflows. Making things that could otherwise be complex and simplifying it and making your job easier. I'm particularly interested in discussing how scientific data management can accelerate innovative approaches like those being pioneered at IDEAYA Biosciences.
Thank you all for those introductions. And let's begin by exploring the current landscape of oncology drug discovery. Mick, could you help us understand the fundamental challenges in developing target therapies for cancer and how synthetic lethality is creating new opportunities in the field?
Okay, so I hear 2 parts about developing and challenges here. So developing targeted therapies for cancer presents several challenges. Cancer exhibits often, genetic diversity across patients within a tumor type, making it difficult to identify universal therapies for these targets. So this is, tumor heterogeneity. That's one challenge that's presented to the field. Despite recent advances in genetic profiling of tumors, a relatively small percentage of patients are eligible for existing therapies based on these profiles. So there is a limited patient eligibility challenge here. That's number 2. Cancer cells develop resistance to therapies over time, diminishing their long-term effectiveness. So this drug resistance is a third challenge. And then I'll just finish up here with the thought that many cancer-driving mutations occur in genes that are challenging to target with current drug technologies. identifying druggable targets is another challenge. So there's just, 4 challenges I've tossed out there. Now, the concept of synthetic lethality, is a promising strategy. Opening new avenues for treatment. Synthetic lethality is a phenomenon, where simultaneous perturbations, talking about a mutation or drug inhibition, simultaneous perturbations of 2 genetic pathways will lead to cell death, while the perturbation of either genetic pathway alone does not lead to cell death. So in simple terms, if you knock out or block a gene, the cell survives. If you knock out or block 2 genes together at the same time, the cell dies. So this concept has important implications for cancer treatment, as it suggests that targeting a gene that is synthetic lethal with a cancer-relevant mutation can kill cancer cells and spare our normal cells. So IDEAYA Biosciences is at the forefront of applying synthetic lethality in this domain of cancer therapy. The synthetic lethal approach involves identifying a synergistic gene pair where synthetic lethality can be exploited. That's our target discovery part. Developing diagnostics to identify patients whose tumors are susceptible to these synthetic lethal strategy. That's our biomarker development part. And then creating drugs that inhibit the target gene in a synthetic lethal pair, effectively killing the cancer cells with the corresponding genetic defect. That gives us, the other part, the therapeutic effect. For instance, IDEAYA is developing therapies targeting MAT2A, PARG, Pol theta, Werner helicase, among others, aiming to treat cancers with specific genetic backgrounds. By leveraging synthetic lethality, researchers aim to overcome limitations of traditional targeted therapies, offering more personalized and effective treatment options for cancer patients.
I think that's really interesting, and clearly synthetic lethality is a compelling approach for precision oncology. I'm really curious, how does it differ from traditional cancer treatment strategies?
Advantages and differences, yeah. So the synthetic lethal approach allows for the targeting of tumor cells harboring specific genetic defects by inhibiting a partner gene, as I mentioned, thereby sparing normal cells. This approach enables the development of therapies that are both effective and selective, minimizing damage to healthy tissues. Traditional cancer treatment, such as chemotherapy and radiation, often lack specificity, affecting both cancerous and healthy cells, which leads to significant side effects. Even some targeted therapies may not distinguish adequately between the tumor and the normal cells, especially when the targets are not unique to cancer cells. In contrast, synthetic lethality exploits specific genetic alterations present only in cancer cells. By identifying and targeting gene pairs where the loss of both leads to cell death, therapies can be developed that selectively kill cancer cells while leaving normal cells unharmed. This precision reduces collateral damage and enhances treatment efficacy. As you can see on the IDEAYA pipeline overview, IDE397 is an example. It's a MAT2A inhibitor. It's targeting tumors with MTAP gene deletion, which is present in approximately 15% of all solid tumors under research now. MTAP, MAT, stands for methionine adenosyl transferase, which is crucial. It's an enzyme responsible for synthesis of S-adenosyl methionine, also known as SAM. MTAP, MTAP stands for methylthioadenosine phosphorylase. Also, an example is IDE161, our PARG inhibitor. It's aimed at tumors with homologous recombination deficiency, also known as HRD, including certain types of breast and ovarian cancer. Another example I'll call out here is IDE275, our Werner helicase inhibitor, focused on tumors exhibiting high microsatellite instability, also known as MSI high, a feature of certain colorectal and endometrial cancers. So by integrating biomarker-driven patient selection with targeted drug development, IDEAYA aims to deliver therapies that are both effective and tailored to individual genetic profiles.
Very good, and thank you for that overview. What would really be interesting now is to focus on IDEAYA's innovative approach and the way you're discovering these new treatments. Would you like to walk us through your vision for accelerating drug discovery and how IDEAYA's platform addresses the challenges?
Sure. IDEAYA Biosciences Harmony platform is a proprietary AI machine learning engine designed to revolutionize the drug discovery process, particularly in the realm of synthetic lethality for precision oncology. By integrating computational tools with structural biology and functional genomics, Harmony aims to accelerate the identification and optimization of novel therapeutic candidates. Traditional drug discovery process is often time-consuming and resource-intensive. IDEAYA's vision with Harmony is to streamline this process in several ways. Utilizing AI and ML to predict compound properties. This is key. It will minimize the number of compounds needed to be synthesized and tested. So here we want to minimize the number of compounds we synthesize. Another aim here is focusing on generating compounds with optimal characteristics such as potency and selectivity and favorable pharmacokinetics. This will enhance the quality of the ideas selected for synthesis. And finally, we want to shorten the timeline from target identification to the investigational new drug application by integrating these computational predictions with experimental validations. So accelerating the timeline from ideas to a drug candidate all the way into the FDA submission of an IND, accelerating that timeline by minimizing synthesis and enhancing qualities of those ideas that we will synthesize. So in summary, this approach allows for a more efficient transition from discovery to clinical development, thereby bringing effective therapies to patients faster.
Yeah, and that's the goal, isn't it? To get the therapies to patients faster. Mick, you might guess, but one aspect of your work that I'm particularly interested in is how IDEAYA has integrated various research informatics technologies into your workflows. Would you like to speak to how partnerships with companies like CDD and Optibrium have enhanced your discovery process?
Yeah, sure. I would love to. Let's dig in a little bit here. So IDEAYA Biosciences has strategically built an integrated advanced informatics platform utilizing the Biochem UDM data model, the CDD Vault, Optibrium StarDrop, and the Harmony machine learning engine into our research workflow to enhance efficiency, data integrity, and decision-making in the drug discovery process. A little bit about each here. Okay, so the Biochem UDM data model serves as a standardized framework for representing chemical compounds and biological assays, facilitating streamless data integration across various systems and studies. This unified model, which is published in the special edition on data standards in the IUPAC journal in 2022, unifies data and supports consistent data interpretation and interoperability. It's considered fair, which is crucial for our collaborative research and regulatory compliance. CDD Vault serves as a secure cloud-based platform for managing our chemical and biological data. It offers key functionality for us, such as chemical registration, for organizing and tracking our chemical entities, assay data management, for storing and analyzing biological assay results, electronic laboratory notebook, also known as an ELN, for documenting experimental procedures and results, inventory management for monitoring compound stocks and usage. I could go on. The CDD Vault platform's RESTful API enables integration with processes, allowing for automated data workflows, real-time collaboration among our research teams. Okay, now let's pivot a bit to Optibrium StarDrop. Optibrium StarDrop enhances our workflow. How does it do that? It does it with data visualization. We leverage interactive data visualization analysis using our plots and charts for structure activity relationships, or our SAR analyses. StarDrop enhances our workflow with these integrations. We have seamless data access with the query interface to AWS Aurora, which is one of our sources of truth for our data. And this enhances and enables our streamlined workflows. So by integrating StarDrop, IDEAYA can make informed decisions on compound progression, reducing the time and cost associated with drug development. Okay, and then finally, IDEAYA's Harmony engine. This accelerates our timeline to an IND. The AI models are used to predict all our tier 1 assays and more before advancing an idea or a virtual compound to a synthesis, thereby reducing time and cost to identify a drug candidate with line of sight into the clinic. So this minimizes our chemical synthesis. Harmony also accelerates our timeline by leveraging a proven and published technology on the AWS well-architected framework and dynamically detecting experimental results upon data capture into the vault, which is synchronized into the cloud, in which we've integrated the query interface right into StarDrop. And this regenerating of prediction models, predictive models, and adapting to new chemotypes in real time allows us, to shrink our IND timeline for our new targets. So there's a synergy between the data model, the Biocommunity M, the CDD Vault, the Optibrium StarDrop, and Harmony. And this allows IDEAYA to, ensure data integrity. The unified data model ensures consistency across all research activities, from ideation all the way through pharmacokinetic studies and all the assays in between. This improves team collaboration. We've centralized data management. We believe in all the data, all the time, everywhere. It's in the data lake, it's in AWS, it's in StarDrop, it's in the Vault. Pick your tools. It's all synchronized, real-time, live. This centralizes our data management and facilitates teamwork among our multidisciplinary teams, internal and external to the organization, including CROs. And again, as you've heard, this helps us accelerate drug discovery. We're using predictive models, whether it's out of the box from Optibrium or it's custom based on our data using Harmony. Data analytics, automated handling of information speeds up the identification of these promising drug candidates. So, in summary, this integrated approach has positioned IDEAYA to effectively tackle the complexities of precision oncology and the synthetic lethal research in which we're conducting today.
Fantastic. I'm really excited to hear more about Optibrium and wonder, Tamsin, if from your perspective, you could go through how the integration between StarDrop and IDEAYA's workflows enhance the synthetic lethality research.
So Mick's described how he's put together a platform of tools here at IDEAYA that's really bringing together multiple vendors and in-house tools to support the specific needs of his team and their workflows. Now, at Optibrium, we've always had a very agnostic approach to integration. For us, it's all about making it easy for scientists to work with their data and the data that's important to them. And this is what enables them to surface the insights that are going to progress their discovery projects. For example, while we have a direct connection to CDD Vault, allowing scientists to retrieve results from their saved queries in CDD Vault, bringing those directly into StarDrop for further analysis. At IDEAYA, we're using StarDrop's query interface to directly connect to their data that's in AWS Aurora. And this is a platform that allows the scientists to save, edit, and share their queries, and bring the data into StarDrop, exactly the data that they want to be working with. We've also integrated with IDEAYA's suite of in-house models in their Harmony platform. And the advantage of that to the scientists is that they have property predictions that are really tailored to the needs of their projects and built on their own project data. These augment the models that we have in StarDrop as well, helping them to prioritize their compound designs. So for us, it's all about ensuring the scientists can access the data and tools that they need in order to be successful with their compound SAR analysis, visualization, and design.
Thank you, Tamsin. That was very helpful for me, and I suspect the audience to learn more about what you're doing there at Optibrium and StarDrop. And Janice, I guess probably we should talk about CDD Vault, and if you wanted to tell us a little bit about how CDD Vault could serve as a foundation that helps IDEAYA connect their experimental data with computational insights.
Yeah, of course. Thanks, Tamsin. Thanks, Mick, for that wonderful description about everything that you do. With CDD Vault, I believe we sit upstream of your data lake, and it's really about, you bring up a really great point about making the data fair, findable, accessible, interoperable, reproducible. We provide this integrated platform where you go from chemical registration and having it be chemically aware, and then now recording assays around that in that integrated platform. And while you might not use all the tools, we do have others that do use all the tools that's offered through CDD Vault. And altogether, everyone's got a piece of the pie where they they're performing some task in their organization, but they need to bring together the whole big picture. And that's why we offer that integrated platform, the ability to put that data in real time, query out that data, or push that data. And like Tamsin was saying about Optarium's tools is we're agnostic, and we're not jealous software. We integrate with other solutions, bringing data in, pushing data out. And at the end of the day, it's to utilize these tools to make life easier so that you can think harder about the science, and you don't have to think harder about that process. You don't have to spend that time there.
Thank you, Janice. And Mick and Tamsin, we've done a deep dive on the science, the IDEAYA pipeline, the workflows, and so on. I think that we should probably spend a little bit of time on the technology platforms, both StarDrop and CDD Vault, to learn a little bit more about the capabilities of each of these systems. Tamsin, would you like to start? Would you tell us a little bit about in the general sense, the key features and benefits of your system, but, we'll try to keep it in the context of oncology research with the understanding that could be more broadly applied.
Absolutely. Let me share my screen. So there we go. Yeah, so StarDrop is really a platform for guiding your decisions, both in the selection of compounds that you already have and the design of new features. So we have our multi-parameter optimization approach called probabilistic scoring to help scientists very quickly target the high-quality compounds for their project, the compounds that are most likely to meet their project objectives, considering all the uncertainty and variability in the data that we're working with in drug discovery. We can also use our chemical space to explore all the opportunities in your project, making selections of compounds that focus on those that are very high quality, but also bringing in diversity when you're selecting compounds to avoid missing important opportunities, perhaps identify a backup strategy. We have suites of very interactive visualization tools, really designed to help you understand the structure-activity relationships in your project. Our unique card view as well is a different way of visualizing and working with the data, helping you to think about compounds, series, and relationships. And it couples very nicely with the SAR analysis algorithms in StarDrop, things like R group analysis, matched molecular pairs, and activity cliff detection to quickly surface some of those key structure-activity relationships. And then, of course, this leads into guiding the next round of compounds, the next round of leads that have that high chance of success. And you can do that with the interactive design capabilities in StarDrop, predicting properties for compounds using the harmony models here at IDEAYA, of course. And StarDrop's glowing molecule helps users to understand the ways that the structure is impacting the properties and where they can make changes to achieve the outcomes that they need. So those are some of the core features that we have in StarDrop.
Very good.
But of course, we have an additional range of optional modules, providing a range of in silico modeling and design capabilities. I'm not going to go into detail on these here, but just say that they fall into four main categories. Our in silico modeling and predictive, for predictive computation, more advanced automated generative chemistry approaches, approaches to 3D design, both ligand-based design and structure-based design, and then some advanced MPO strategies, thinking about the development and analysis of those MPO scoring profiles. Now, the real focus today is, of course, the flexible integration and customization, helping teams like IDEAYA get the most from their research and infrastructure, and providing that very intuitive access to everyone through the integrations with other platforms and in-house tools. And some examples of those are shown on the right of this slide, and in particular, the custom models that here are in the Harmony platform. And then if we think about the custom Python scripting, Stardrop links with a wide range of third-party platforms. I've listed here some of the different approaches, and I wanted to just highlight the data management and informatics platforms, including our direct link to CDD Vault. And then another approach here is the query interface, which is providing the team here with that seamless access to their most current data. Here it's stored in the Aurora, but it can be stored in data sources ranging from flat files through web services to relational databases, and provides this very user-friendly tool for creating, sharing, and running these queries. And importantly, it also refreshes the analysis so that when a scientist picks up Stardrop after being away for a couple of days, rerun that query, and it will highlight for them what's new, what's changed, what the rest of the team has been working on while they were away. So that's a very quick and high-level summary of some of the capabilities that we have in Stardrop and how we're trying to support teams like IDEAYA's.
Thank you for that, Tamsin. Janice, I wonder if you could share how CDD Vault's capabilities support the unique data management needs of precision oncology and beyond.
Right. So I too will give you sort of this high-level overview of CDD Vault, where we start from registration and registering securely your assets, your IP. And Mick mentioned his biochem UDM model. And what we have the ability to do is configure our system that's been around for 21 plus years and kind of applying, your sort of business rules to it and having the agency to do that. And you start in the lower left-hand corner with that registration, and those are your chemical compounds, but we also allow you to register biologics, macromolecules. You will record data for those molecules, those assets, and what we can do is now take projects to allow you to collaborate with your data. We're putting this data in real time, but we may not want to share all of the data. So projects are going to be a way of now organizing that data cleanly, but also controlling who has access to that data. And above that, if you're going through and registering these assets, let's keep inventory of those things that we're registering into the vault. And of course, Mick did mention the electronic laboratory notebook. This is where we put our unstructured data. With the assays that sit above the ELN on the right-hand side, you could imagine that in making the data fair, you would like to create an assay catalog of the experimental data, the actual experiments that you're running for those substances that you're testing, and creating this assay catalog to better organize that data, making it fair. Of course, there's the automation aspect of it, going from your instruments, putting that into CD Vault, going back out, and that makes use of the RESTful API. And then additionally, complementing all of that, and as we mentioned before, well, IDEAYA may not utilize all of these tools. There are companies that are bootstrapping, and let's paint that big picture. Let's take our SAR, go and make quick go, no-go decisions, save us time. We have visualization within CDD Vault. Let's analyze the curves, the data that we're getting from our machines in many different ways. And then we too also have our AI models within for deep learning similarity searches, bioisosphere suggestions. We just recently announced a partnership with NVIDIA, and that's to, now actually have AlphaFold 2 and DiffDoc within our platform, and easily, now take that data and model that data for more powerful tools within this system.
Thank you, Janice. And Mick, I'm just thinking about your background and your experience. I think it would be helpful for us, the audience, to understand your perspective as, I would say, both from the user perspective and what my IT friends call as a solution architect, someone that is a designer behind the system. What features or integration points have you found most valuable as you build up the system that IDEAYA Research uses?
A good question, Eric. So, as you can tell, we're a cancer oncology research development company. And at this time, we're aiming to go commercial. At the heart, we're scientists. Yes, we have some computer science skills. We're not a software company, so we're strong in science. We want to partner with the folks at the CD Vault, with the folks at Obtibrium, that give us the technology that's easy to use, it's powerful, that works. There's some devil in the details. It's worth sorting those things out and getting those things done. We like to build things once and build it right and let it run forever. And we've built a platform that does this. So integrating platforms that leverage the technologies we've touched on today, using the BioChem data model, the CD Vault, the Obtibrium StarDrop, and the Harmony engine, has proven to streamline the drug discovery process by unifying data, enhancing collaboration, enabling faster data-driven decisions. This reduces errors, it accelerates candidate selection, and improves our overall R&D efficiency.
It's good. Efficiency is improving efficiency is always the key point here, especially in drug discovery. And what I'd like to do is transition back a little bit to the science. And I think all these systems, all these technologies are incredibly interesting and useful, but it's always good to demonstrate some true value, right? So they call return on investment. I'd love to hear, if you could share some examples of how these platforms have helped you with maybe specific use cases or programs, to the extent you can talk about those, and the benefit of having those systems available to you for those programs.
Okay, well, as you've heard, IDEAYA leverages its Harmony platform to rapidly address key challenges in compound solubility, for example, and metabolic stability in all tier 1 assays. I'm talking about anything involving binding, inhibition, induction, oxidation, activation, any sort of biochemical or cellular assays, pharmacokinetic studies, permeability, all sorts of physical chemical properties, stability of all sorts, including metabolic stability, high-tech experiments coming out of our service plasmid resonance groups, toxicity experiments. Harmony leverages all the data publicly and privately to address these key challenges in predicting these important properties before we synthesize compounds. This approach has significantly accelerated our efforts across multiple programs. Going back to our pipeline, I'm talking about PAR-G, PRMT5, the CAT6/7 program. As a result, IDEAYA now addresses high-quality candidates with improved drug-like properties with fewer design, make, test, analyze cycles.
Yeah. Have you thought about, the big push, especially in constrained funding environments, to talk about metrics and return on investment and measurable improvements based on various technologies. Is there anything you can share with us? Have you done the sort of back the envelope calculations on how implementing these types of technologies have improved efficiency or accuracy, or frankly, even maybe some cost savings for freeing up time of scientists to do other things?
Yeah, sure. We've done some back of the envelope calculations. That was in the early days when we started the proof of concept. Once the platform was launched, we have implemented monitoring and tracking. And so we have some real hard data on this. And we find that Harmony primarily saves time and secondarily reduced costs. But again, we're trying to shrink the IND timeline. Yes, we'd like to save money. Money's not the problem here. It's really the time from idea to the clinic. And in order to enable successful clinical trials to deliver these benefits to the patients sooner, we had Randy from one of our clinical trials come into our office, sit right here on the couch, all hands meeting, thanking us for bringing our medicine into a phase 1 trial that he participated in. Some of the standards of care are not very attractive for people with uveal melanoma. I'm talking about cancer on the eyeball. We're talking about radiation of the eye. It doesn't differentiate between cancer cells and healthy cells. If radiation doesn't reduce the cancer enough before side effects and toxicity comes in, you're going to the second step, which is enucleation of an eyeball. He was thanking us for saving his eyeball. That's really motivating for all of our researchers and our scientists. So being able to put a medicine into the clinic that's safe and save, patients from cancer, basically, in this case, Randy saving his eyeball. I can't think of a better metric than that. So, yeah, we're primarily saving time to the clinic. We're secondarily reducing costs in order to deliver these benefits into the patients in trials sooner. We hope to launch darovasertib in the next 12 to 18 months. This will make the medicine widely available to patients in need. We hope all of our medicines in the pipeline can deliver value to patients. And our internal metrics on this technology that we've talked about here today indicate that our machine learning engine enables us to reach the clinic about 10 to 30% faster, depending on the synthetic lethal targets and the competitive landscape.
I couldn't agree with you more about the primary metric should be medicines to patients who need those medicines. And I go back to my past experience a long time ago with the company, and one of the most motivating mission statements I'd ever heard was simple words: "The patient is waiting." And that really drove us to try and work harder, faster, more efficient.
Hear that?
We got just a few more minutes, but I think it's okay if we have a few final thoughts before the question and answer session. And this would be a good opportunity for each of you to share, a brief final thought on the future of these integrated approaches, some things that you might not have been able to get out during my questions, and we'll go from there. So, Mick, would you like to start?
Yeah, so just, briefly about the vision of the future of synthetic lethality and the integrated drug discovery approach we've talked about here today. So for those that maybe didn't catch the whole seminar, they're just jumping in somewhere here, they're too lazy, didn't read, here's a nice summary. The future of synthetic lethality and integrated drug discovery approaches lie in delivering increasingly precise, effective, personalized cancer therapies. Synthetic lethality will expand beyond current targets, driven by deeper genetic insights and novel biomarker discovery. This should enable treatments for previously untreatable tumors. This integrated discovery platform we've talked about here today, it combines artificial intelligence, machine learning, multiple easy-to-use platforms and vendors, as you've heard from Janice at the CDD Vault and Tamsin from Optibrium. This has enabled us to automate and integrate workflows and unify our data systems. At the end of the day, I'm an informatics guy. At IDEAYA, we have 131 employees. In informatics, we're an N of 2. How do we support a pipeline like this? Well, we've just described it to you. We're publicly traded on the NASDAQ. We got a market cap of about $2 billion. This works. I can't say enough about our partners at the Vault and at Optibrium. These tools work. They're robust. They're rock solid. We've pressure tested them. We've challenged them to make it better. They're easy to use. Our scientists understand them intuitively. They can start using these tools on the first day. We launched them live in less than a year after doing a proof of concept. So that's super exciting. So this integrated discovery platform combining AI and ML in our workflows and our unified data model in our systems, this is accelerating our drug development cycles. This is reducing our costs as well, and it's improving our candidate quality even before we make the molecule. So together, collectively, you've heard from Janice and Tamsin and myself today about how these innovations will transform oncology by making targeted therapies faster to develop, more affordable, accessible to broader patient populations sooner than later.
Very good. Tamsin, would you like to add anything?
Oh, sure, I can do that. Yes. So I'd say across the industry, we're seeing more distributed project teams, and that really increases the need for scientists to be able to have that ready access to the data that they need to work with, and to avoid silos of data. And I'd say we're also no longer in the days of a single software vendor, a platform from a single vendor. Instead, I think we're going to see more situations like the one that Mick has described and put in place here, this innovative platform really designed to meet the needs of his team, bringing together the right tools to do the right jobs. Okay, and so, helping to support the scientists and their research, both from commercial and in-house sources, with the goal, as you said, of meeting the needs of the patients and getting there faster.
Good. And Janice?
Yeah, I'd say, just to add on to that, using these different software platforms that do nicely integrate with one another, that do play nice with one another, and the people within them play nice with one another as well. I've gotten to know Tamsin over the years, gotten to know Mick, and Mick brings up a great point in that he's pushed us. We've had this infrastructure for over 21 years now, and there are demands of the industry that change, that evolve over time, and we change with our customers, we change with them, we evolve so that they can do the things that they need to do. So, and really, I think Mick does a really great job here to describe how he takes advantage of our solution, that we have this very intuitive user interface. And they're two people that, now train a whole site. I just wonder if there are other benefits that you see from CDD Vault and working with CDD Vault and all of this, and now handling all of those complex structure activity relationship data that is in your vault, and how it applies to that UDM model.
Yeah, I can riff off that. We can go on and on. One capability in the vault that is near and dear to my heart is compound registration. I mean, at the core, I'm a chemist. I understand structure. I understand enhanced stereochemistry. I understand this is the language of the chemists. So the vault is able to register compounds and identify duplicates, including Boolean logic on stereocenters, atropisomers, mixtures, and so much more. We're expanding into ADCs. This sort of expertise in a tool like the Vault is extremely valuable. We love things that work, and the Vault works. And another thing we really like is the RESTful application programming interfaces, the APIs. They're robust. We have pressure tested this to the max. We have integrated into these APIs. It's robust. We've built this infrastructure, as you saw on one of the slides, and it's running 24/7, 365. And it's continuously integrating new experiments being developed by data generators, being captured into the vault, synchronizing to the cloud. We're spinning up, new data sets in the Stardrop all the time. Harmony is dynamically training on data sets. And I'm sure just during this webinar, new data has come in and it's already appearing in the vault and it's already appearing in Stardrop.
Much as I'd like to continue on and on, we really need to get to the questions in fairness to the audience. But before I do that, let me say a few things about a poll that we're taking. I'd like to draw your attention to a poll that's now appearing on your screen. And if any of these companies and the technologies we've discussed today look interesting to you, please take a moment to vote. You can indicate whether you'd like a customized demo or if you'd prefer for us to just send you some resources by email. There's the option for either. Please let us know. So let's move on to questions, if that's all right with the panel. Here's one here. Start at the top with one for Mick. He says, what are the key criteria you use when selecting and prioritizing synthetic lethality targets, especially given the complex genetic landscape of different cancer types? I know you touched on this a bit, but maybe you want to, give a bit more detail on that topic.
Yep, a good question. A little detailed, quick summary as well. Key criteria include tumor-specific genetic dependencies, prevalence of the targetable mutation across the cancers, availability of predictive biomarkers, and druggability of the target. This leads us to potential therapeutic selectivity to minimize the toxicity in normal cells. Yeah, that's what I'd say.
Very good. Now I'm going to just try and partition these across each of the panelists. So let's try to be fair here. So for Tamsin, there's a question. It looks like it would be right up your alley. What does an integration like the one that's been described today between StarDrop and all the other tools involve? In other words, I think I'll just paraphrase. How hard is it to do what Mick's described? What's your thinking about the amount of time, energy, resource to do these types of things?
Oh, of course. Yes, that's an easy one to answer. We provide the tools for organizations to do a lot of this themselves if it's fairly straightforward. And I think that's largely what Mick did here at IDEAYA. But every organization's data structure is different. So we also have a team that would meet with a customer, learn about what your specific needs are, and then work with you in order to be able to customize that integration to meet your needs.
Very good. And for Janice, I'm going to pick a question here that actually is part of the CDD name that gets buried under the C, collaboration. What specific features in CDD Vault are designed to facilitate collaboration between computational chemists, and I would say synthetic chemists as well, or bench chemists, and biologists working on targets? In other words, how do you facilitate collaborations? Yeah.
Yeah, well, with the integrated platform, we allow you to, own that piece of the puzzle and put it all together. There's a component for chemists to register their chemical compounds, a place for biologists to store their assay data and design protocols, we call them protocols, to house that data, and basically assemble it all together with the back end that we built in terms of that approach to it. And we make it so that you can organize this through projects and bring those projects together, and also, secure it in such a way that all the projects don't have to be shared and it's on a need to know basis. So you can collaborate. You can now also collaborate in ways that you manage in terms of managing permissions.
Very good. So I've just got a little chat message here. We can go one more round. So let's do it one more time. Let's start with Mick. Hey, Mick, could you elaborate on, well, what's this question? Let me read. Could you elaborate on how your team balances the computational predictions with the experimental validation when evaluating the potential synthetic lethality of targets? In other words, I think summarize, how do you validate the models, and do you use these systems to help you to validate models?
Sure, yeah. Briefly, we use computational predictions to prioritize our targets based on the genetic context and druggability, and then we validate them experimentally through the functional genomics, biochemical assays, and in vivo models to ensure the biological relevance and the therapeutic potential. And I would add to that, some of the main challenges were data compatibility, user training, systems integration, which we've heard about today. We overcame them by adopting a data standard, providing cross-functional training, and leveraging the APIs on the Vault to ensure both smooth communication between platforms for a unified workflow.
Can multiple scientists work together at the same time in StarDrop?
Great question. At the moment, not directly. I think you're talking here about being able to collaborate in a single data set. So there's some limited things that you can do, but we're really excited about later this year. We're going, in our next release of StarDrop, we're going to be making it a truly collaborative platform, allowing everybody to work together interactively and see those changes that their colleagues make, any notes or annotations in the data, see those immediately. So that's really going to help to streamline and speed up the research and the insights that the scientists are going to get from their analysis.
Very good. And for you, Janice, this is a big, complex question, but I think if you could just give a few words and then we could direct people to follow up with you later. How does CDD Vault's data security approach address the confidentiality concerns that arise when sharing sensitive data across different organizations and research sites?
Right, and that's great. It is through the agency of, deciding who can be a member of your vault, and then we've got layers of security that you could add on to that. So we can do it that way, or you don't invite someone into the vault, and it's just the ease of pushing a button to generate a report from CDD Vault and send that out. You can do it in many different ways.
I want to thank Dr. Michael Kapler for sharing IDEAYA's vision and approach, Dr. Tamsin Mansley for illuminating how Optibrium's computational tools support this important work, and Janice Darlington for explaining how CDD's data management solutions provide the foundation that connects experimental and computational insights. To our audience, most importantly, thank you for joining us today. The recording of this webinar will be available on our website, along with additional resources on the topics. If you're interested in learning more about how CDD Vault, StarDrop, or IDEAYA approaches could benefit your organization, representatives from all three companies will be available for follow-up conversations. Thank you for your participation.